Hello, this is Amin Alirezaylavasani. I'm a master's student of molecular medicine. I perform my master thesis at Oxenic Research Group, and I'm supervised by Dr. Valentin Oxenic and my co-supervisor Sergio Castanet. All cells function using the proteins that are encoded by genes. When essential genes are mutated, the consequence might be serious. DNA damage occurs every day due to exogenous factors such as UV radiation or endogenous factors like reactive free radicals generated during metabolism. And there are different types of DNA lesions, but the most severe one is DNA double-strand break. To preserve homeostasis, our DNA should be repaired and resist changes. There are different pathways dealing with DNA double-strand break, like non-homologous engineering repair, homologous recombination, and single-strand annealing. But non-homologous engineering repair, or NHEJ, is the major pathway dealing with DNA double-strand break in mammalian cells, and it is active all over the cell cycle. Consists of core factors and accessory factors. Core factors are essential for any kind of DNA double-strand break repair and include Q7080, ligase 4, XRCC4, and XLF. On the other hand, accessory factors are required for more complex DNA double-strand break repair and include DNA PKCs, PAX, MRI, and Artemis. After double-strand break induction, NHEJ recognizes and processes double-strand break. Initially, Q70 and 80 form a heterodimer and associate with double-strand break and facilitates the recruitment of downstream factors. Protein kinase DNA PKCs functions with Q heterodimer and phosphorylates most of the NHEJ factors. Following, the nuclease Artemis, with its 3' to 5' exonuclease activity, open DNA hairpins and makes DNA strands prepare for DNA ligation, and then ligase 4 ligates broken DNA ends. Meanwhile, XRCC4 makes a connection between ligase 4 and DNA ends to increase ligation activity. XLF makes a connection between XRCC4 and other NHEJ factors. PAX stabilizes the whole NHEJ machinery. And finally, MRI regulates NHEJ activity during S and G2 phases. The cooperation between NHEJ factors results in fixed DNA. NHEJ is required for VDJ recombination. VDJ recombination is a process that is required for rearrangement of genes encoding heavy chain and light chain of B-cell receptor, or BCR, as well as rearrangement of genes encoding beta chain and alpha chain of T-cell receptor, or TCR. VDJ recombination, during VDJ recombination process, DNA double-strand breaks are induced, and then a selected diversity or D gene segment connects to a selected joining or J gene segments and form DJ complex, and then a selected variable or V gene segments connects to DJ complex. When we look at the lymphocyte development, we see that in bone marrow, hematopoietic stem cells produce immature B cells through B cell development process, and then immature B cells migrate to spleen and locate there. Meanwhile, hematopoietic stem cells produce progenitor T cells. They travel through blood to thymus, and in the thymus, they produce T cells through T cell development process. And then T cells migrate to spleen as well. VDJ recombination is required for both B and T cell development. And as it mentioned before, NHEJ is required for VDJ recombination. So we can say that successful NHEJ leads to successful VDJ recombination that leads to successful B and T cell development. In contrast, deficient NHEJ leads to VDJ recombination failure that results in no B and T cell development that leads to immunodeficiencies. It has been reported that there are 6 million people throughout the world that are dealing with immunodeficiencies, but noticeably 60 to 90% of them are undiagnosed. The reason is incomplete diagnosis and therapy approach. So we study NHEJ and the roles of NHEJ factors in B and T cell development in knockout mice to understand NHEJ machinery and B and T cell development related disorders. I should mention that mice lacking XLF are live-born with modest immunodeficiency. In addition, mice lacking PAX or MRI are live-born with normal lymphocyte development. Mice with single inactivation of DNA PKCS are live-born, but they have severe combined immunodeficiency, means they have no B and T cells. But the point here is that some DNA repair factors have overlapping roles, which makes it more challenging to study them. The solution is genetic interaction study. It reveals functional relationships between genes. For example, if we imagine gene A and gene B, when they are both present, mice is alive. Single inactivation of gene A or gene B poses no serious phenotype, but inactivation of both genes leads to lethality. It means that gene A and gene B interact genetically. Studying the genetic interaction between XLF and PAX revealed that single inactivation of XLF or PAX poses no serious phenotype, but mice lacking both XLF and PAX are embryonically lethal. Inactivation of TRP53 gene rescues lethality of XLF-PAX double deficient mice, and studying XLF-PAX TRP53 deficient mice shows those mice poses B and T cell development deficiencies. So I hypothesize that lack of B and T cells in XLF-PAX TRP53 deficient mice is rooted in bone marrow and thymus, caused by NHEJ blockage at the early stage of lymphocyte development. Moreover, single inactivation of MRI poses no abnormal phenotype. Mice lacking DNA PKCS, on the other hand, are live-born, but they are immunodeficient. The question is whether MRI and DNA PKCS interact genetically. So I hypothesize that MRI and DNA PKCS interact genetically, including aspects of B and T cell development. Similarly, single inactivation of MRI and PAX poses no abnormal phenotype, but it is unknown whether MRI and PAX interact genetically. I hypothesize that MRI and PAX interact genetically, including aspects of B and T cell development. Sorry. To test my first hypothesis, I analyzed 54 mice generated from the parents that both were XLF-, PAX +-, and TRP53 +-. I found that XLF-PAX TRP53 deficient mice are visually smaller and lighter when compared to single knockout and wild-type controls. In addition, the counts of splenocytes is severely reduced in XLF-PAX TRP53 deficient mice. Similarly, the counts of thymocytes is severely decreased in XLF-PAX TRP53 deficient mice in comparison to single knockout and wild-type controls. Noteworthy, the counts of bone marrow cells is significantly low in XLF-PAX TRP53 deficient mice. To screen B and T cell development in mice, I isolated spleen, thymus, and bone marrow. Splenocytes were immunostained using anti-CD19 and anti-CD3 to show the frequency of B and T cells, in addition to anti-CD4 and anti-CD8 to show the frequency of T cell subsets in a spleen. Moreover, thymocytes were marked using anti-CD4 and anti-CD8 to show the T cell development. Finally, bone marrow cells were stained using anti-B220, anti-CD43, and anti-IgM to show B cell development in bone marrow. I found that the proportion of splenic B and T cells, means B and T cells that were harvested from spleen. The proportion of splenic B and T cells is severely reduced in XLF-PAX TRP53 deficient mice. Here's the flow cytometry analysis of splenocytes using CD3 and CD19. Statistical analysis prove that the frequency of B and T cells is severely reduced in a spleen of XLF-PAX TRP53 deficient mice. Here is the flow cytometry analysis of splenocytes using CD8 and CD4 markers. And the statistics demonstrated that the frequency of a splenic CD4 positive and a splenic CD8 positive is severely reduced in XLF-PAX TRP53 deficient mice. Analysis of thymocytes using CD8 and CD4 markers shows that thymic CD4 positive cells, thymic CD8 positive cells, and thymic double positive T cells. The frequency is severely reduced in XLF factor 53 deficient mice in comparison to single knockout and wild-type controls. Finally, flow cytometry analysis of bone marrow cells using B220, CD43, and IgM shows that the frequency of B220 positive and IgM positive cells are decreased in the bone marrow of XLF CR53 deficient mice, while the proportion of Pro-B cells is increased. So in conclusion, XLF CR53 mice poses deficient B and T cell development, and B cell development is arrested at Pro-B cell stage in bone marrow of XLF CR53 deficient mice. To test my second hypothesis, I analyzed 66 mice that were generated from the parents, both heterozygote for MRI and DNA PKCs genes. Noticeably, I did not find any MRI DNA PKCs deficient mice. This led me to plan another experiment. Then I examined embryos at embryonic day 14.5, and interestingly, I found 2 embryos that were smaller and lighter when compared to other embryos. Genotyping PCR later revealed that they were both MRI DNA PKCs deficient. So in conclusion, the number of analyzed mice may be too low to make a solid conclusion, and total 100 to 400 mice need to be analyzed in the future, but it may suggest that MRI DNA PKCs deficient mice are embryonically lethal due to the genetic interaction between MRI and DNA PKCs genes. In order to test my third hypothesis, I analyzed 60 mice that were generated from the parents that both were heterozygote for MRI and PAX genes. I found that MRI PAX deficient mice have normal weight and possess normal proportion of splenocytes, thymocytes, and bone marrow cells. I followed the same pattern of studying for MRI PAX deficient mice, and on this slide, it is not required to follow the statistics. I just put them and mentioned them to say that after analysis, I found that MRI PAX deficient mice have normal B and T cell development. So in conclusion, there is no genetic interaction between MRI and PAX genes in vivo. So I have validated my first and my second hypothesis, but the third hypothesis appeared to be wrong. As future directions, we collected brain samples from MRI DNA PKCs deficient embryos, in addition to single knockout and wild-type embryos. I hypothesized that massive apoptosis in central nervous system, in addition to increased genomic instability, is the cause of lethality of MRI DNA PKCs deficient mice. So hematoxylin and eosin staining can be used to screen the apoptotic and necrotic bodies in the brain tissue, in addition to T-FISH assay to see the level of genomic instability. At the end, I want to thank my supervisor, Dr. Valentin Okseny, for his continuously guidance and support. I thank my co-supervisor, Sergio Castaneda, for his numerous theoretical and practical guidance. And I want to thank my colleagues, Shingdon and Kalai at Okseny's research group. Thank you so much for your attention.